And that's how we can think about potentiating drug effects with combinations of agents by knowing the pathways. And that becomes important to treat cancer and various cardiovascular diseases. Now, pathways are often more complicated than you think initially. When you make nitric oxide, what it really prefers to do is activate soluble cyclase to make cyclic GMP. That's what it wants to do. However, it's pulled off in other directions. There are other things coming after it. It'll get oxidized to nitrite. Initially thought to be inert, benign metabolites. Not true anymore. They are pools of potential NO donors. They can be converted back into nitric oxide. They can form a reaction with other transition metals to form nitrosyls. There are metalloenzymes that are regulated and inhibited by NO. It prefers to interact with iron, but it can interact with zinc and other transition metals as well. It forms complexes with thiol groups, cysteine thiols, in a variety of proteins, hundreds of proteins. And when these proteins are nitrosated or nitrosylated, their activity can change. Sometimes they're receptors, sometimes they're transcription factors, sometimes they're caspases, there are a variety of things. Another potential signaling pathway for NO. A very important reaction is the interaction of nitric oxide with superoxide anion. This reaction is almost diffusion limited. What does that mean? It means it's so fast that that's what will happen. If they're both in the same environment at the same time, you make peroxynitrite. So you never want them in the same place at the same time because peroxynitrite is very toxic and destroys lots of macromolecules, RNA, DNA, proteins, etc. One of the footprints of peroxynitrite formation, and it's difficult to measure it, is its downstream effects, and that's the nitration of tyrosyl residues in proteins. So when you see protein tyrosine nitration, it's an index that you made peroxynitrite, and you've had another inflammatory insult. With inflammation, you jack up in NOS2 production, as I told you, through NF-kappa B, but you also jack up peroxynitrite formation, superoxide formation through NAPH oxidase and xanthine oxidase as well. So they're biomarkers. Now, if we could block peroxynitrite formation, perhaps we could come up with another approach for inflammatory disease therapy. Nobody's been successful with that. The inhibitors have not turned out to be very good. If the reaction were reversible for protein nitration, that would be another approach to treating inflammatory diseases. We thought there was such an enzyme, and we pushed on it pretty hard for a couple of years, but the data are not that convincing. We are concerned about it. I would like to think that it is reversible because it would be a lovely place to go for discovering some new drugs. But that's not been the case. Okay, let's turn to endothelial dysfunction. Patients with hypertension, atherosclerosis, diabetes, tobacco use, and probably obesity have blood vessels that do not make sufficient quantities of nitric oxide. Your blood vessels are compromised, your blood flow is compromised to your tissues, you're not delivering adequate oxygen and nutrients. For a variety of reasons. All of these diseases are associated with another metabolite of arginine that's a competitive inhibitor at the catalytic site of NOS. To degrade proteins, one of the pathways is methylation of arginine. And when you form asymmetric dimethylarginine, you have a competitive inhibitor at the arginine catalytic site with NOS. You can overcome that competitive inhibitor by more arginine. You also have in these diseases reactive oxygen species, which are going to oxidize the cofactors, tetrahydrobiopterins say, to dihydro. And when those cofactors are depleted or oxidized, the enzyme becomes uncoupled. It no longer makes NO, and in fact, it makes superoxide anion, the worst thing you can do, because the superoxide now is going to scavenge what NO you do have. So for a variety of reasons, all of these are associated with reactive species and are influencing NO production or activity. And how do we overcome that? Well, I think there might be ways by regulating redox chemistry, perhaps with antioxidants, perhaps by supplementing arginine in the diet to overcome the inhibition by asymmetric dimethylarginine. But there are lots of arginine products out in the market, very, very few clinical trials, and I think we need to do more clinical studies to really prove to ourselves that they're really efficacious or not, and they're missing. And I help advise a number of companies about some of these nutritional products, but I also try to convince them, and they promise they'll do clinical trials, but usually they don't because they don't want to waste the money. And I keep saying, if you do the studies and they're positive, you'll sell more product. But they're hard to convince sometimes. Okay, let's think about other potential applications for this broad field. And this is my David Letterman list. I've got 2 slides of diseases. I don't work with all of these. I work with some of them. But I tend to follow many of them because I think they're interesting and have a lot of potential as to where the field is going to go in the next decade or so. We know that nitric oxide is a neurotransmitter. There are nerves in the brain, the GI tract, in the corpus cavernosum of the penis that are nitrinergic nerves that release nitric oxide as their neurotransmitter. If you knock out NOS1 in a mouse, it induces stroke. The stroke is smaller. If you knock out NOS3 and induces stroke, the stroke is bigger. If you do a double knockout NOS1 and NOS3 and put the mouse in the water maze test, they can't find the float. They have a defect in memory. So there are some interesting effects of NO and perhaps cyclic G in the brain, but we don't understand all the details yet. And that's because the brain is a very complicated, heterogeneous box where it's difficult to do a lot of biochemistry. We know it plays a role in fluid production in the eye, retinal degeneration. It plays a role in pulmonary hypertension. Babies in utero don't need to perfuse their lungs. They perfuse the placenta. That's where they get their oxygen from the mother's blood. When they're born prematurely at 1 or 2 kilos, their lungs are not developed. The pulmonary vessels are vasoconstricted. They shunt blood right to left because of the pulmonary hypertension. They shunt blood through the patent foramen and ductus arteriosus. They're blue babies. If you give them low concentrations of nasal nitric oxide, they dilate the pulmonary vessels. Their shunting diminishes, their oxygenation improves. It's really been a marvelous addition. And now, in fact, in many hospitals, it's being done in combination with sildenafil Viagra to potentiate the effects of the inhaled NO. It has a role in angiogenesis, in asthma, with either infection or allergy. You induce NOS2 in the lung to make more NO. Therefore, there's more nitric oxide in the exhaled air. The doctor can follow exhaled air, and as it goes up, they know that the asthma is getting out of control, and they have to treat the patient with antibiotics or steroids or whatever. So it's been useful as a monitor of asthma therapy. We know that it inhibits platelet aggregation. I have a theory that patients who have bypass surgery on the pump have microaggregates of platelets. Therefore, they have microemboli. We're infarcting off pieces of the kidney and brain of all these patients that have open heart surgery. What we need to do is inhibit platelet aggregation as the blood goes through the pump. And I think NO and prostacycline and some other additions would be useful. But no studies have been done carefully to prove that, which is sort of surprising. We know it regulates gene expression. With microarrays, we can see 12 or 15 genes go up and about a half a dozen or so go down. So it does regulate gene expression. We don't know the mechanism. Is it NO directly? Is it NO through cyclic G? Is it through a protein kinase? Are there binding proteins for NO or cyclic G? We don't know. But it does happen. And I think it plays a role in stem cell differentiation as well as cancer cell proliferation and growth. We can modify NO production in embryonic stem cells, and make them become myocardial cells or neuronal cells in culture. I think that's going to be important someday as we understand all of the regulatory growth factors of stem cells as we re-engineer tissues. I think there could be a day, 20, 30 years from now, where they'll take a biopsy from you and get your stem cells and grow them up in the lab, and you won't need a liver or a heart or a kidney anymore. They'll engineer that tissue in the laboratory and give it back to you. And I think that'll come. It's just a matter of time. So it's been fun to see this all happen. And as I told some of your faculty earlier, I think you've got to think outside of the box sometime and just be creative and imaginative, and sometimes even crazy, because that's what it takes to sort of get somewhere and do something interesting. Thank you very much.